Good day, students. In this lecture, we will be discussing the cell division type meiosis, that is the third type of cell division. Previously, we have discussed about amitosis and mitosis types of cell division. So, let us talk about the third type, that is meiosis. Meiosis term was coined by two scientists, that is Farmer and Moore. As we have studied during mitosis, that this mitosis division occurs during the body growth, repair, replacement of cell. But if we talk about meiosis, this division occurs only once in the life cycle of an organism, and that is during gamete formation. So it occurs only during the gamete formation. And these gametes participate in the sexual reproduction process. One more important point about meiosis is that it involves two divisions during which a diploid cell divides two times to form four daughter cells which are haploid. Let me explain you what is diploid and haploid first. Diploid cell has two set of chromosomes. So, diploid cells are those cells which have two set of chromosomes and they are represented by 2n, where 2 represents two sets, whereas n represents the types of chromosome. Whereas a haploid cell has only single set of chromosome and it is represented by alphabet N only, which represents only one copy of the different types of chromosome. Remember one thing, that each organism has a specific number of chromosomes. Say for example, if we talk about human beings, they have 23 types of chromosomes. So if we talk about a diploid human cell which is present in the normal body cells, they have these 23 types of chromosome in two sets. So each type of chromosome will be present in two copies. But if we talk about a haploid cell like gamete, the gamete cell like sperm or ovum or the egg cell will have only single copy of each type of chromosome. So it will have only 23 chromosomes, whereas a diploid human cell will have two copies of each. So it will have 46 chromosomes in total. So now we can understand meiosis properly. Meiosis involves double division. So, in this meiosis process, a diploid cell divides two times and this leads to formation of four daughter cells which are haploid. So, the daughter cells are identical to each other, but they are not identical to the parent cell, because in the parent cell there are two set of chromosomes, whereas in the daughter cells there are only single set of chromosomes. For example, if we say that a parent cell is having four chromosomes, total number of chromosomes is say 4. Okay. Now if it divides by meiosis, it will lead to formation of four daughter cells, but each of the daughter cell will have only half the number of chromosomes, that is, it will have only two chromosomes. So the four daughter cells are identical to each other in the sense that they are having two chromosomes each, but they are different from the parent cell in sense that the parent cell was having four chromosomes, but the daughter cells, all the four daughter cells are having two chromosomes each. So the daughter cells are not identical to the parent cell if they have formed by meiosis process. Now where meiosis can be studied? It can be studied in anthers of the unopened flowers in plants and in the testes of grasshopper. Grasshopper is an example of animal. So we can study meiosis in animals as well as in plants. Remember that the anthers and the testes represent the reproductive organs which lead to formation of the gametes which are haploid. Now, as I told you, we will be discussing the brief outline of meiosis and we'll discuss it in detail later on. Meiosis is divided into two parts, and in between these two divisions, interphase can be or cannot be present. So it comprises of two divisions, short or no interface between them. Remember, interphase is present between the two divisions, that is meiosis 1 and meiosis 2. Then that interphase will comprise only of G1 phase and G2 phase. It will not contain S phase during which DNA replication occurs. So it means that if there is a interface between meiosis 1 and meiosis 2, there will be no DNA replication. Only there will be growth 1 and growth 2 phases. So DNA will not divide again. DNA will divide only once at the start of cell cycle during the initial interphase. DNA will not replicate during the interphase between the two divisions of meiosis. Meiosis in turn is also called as the reduction division because the cell changes from diploid stage to haploid. Meiosis 2 is known as the equational division. Meiosis 2 is known as the equational division because in this stage the haploid state of the daughter cells is maintained. So from haploid daughter cells, further haploid daughter cells are formed. Because in meiosis 2, the daughter cells which are formed during meiosis 1 again divide. So haploid state is maintained. The haploid daughter cells further divide by haploid meiosis 2 and form haploid daughter cells. Meiosis 1 or first meiosis occurs in 4 steps, and the 4 steps are same as that of the mitosis, that is prophase 1, metaphase 1. Remember, the steps are same as that of the mitosis, anaphase 1 and telophase 1. The prophase 1 is the longest phase of meiosis and it is further divided into 5 stages, that is leptotene, zygotene, pegitine, diplotene and dikinesis. The stages of prophase 1 are leptotene, zygotene, fecitine, diplotin, and dikinesis. After completion of the prophase 1 by these 5 steps, the next step is metaphase 1, then anaphase 1, and finally telophase 1. Similarly, as like meiosis 1, meiosis 2 also occurs in the 4 steps, and the steps are same as that of mitosis, or we can say meiosis 1. And it is very similar to that of mitosis. Although meiosis 1 is different from mitosis, but it contains same steps as the main steps of meiosis 1 are same as that of mitosis, but it is quite different from mitosis process, because in this stage the diploid parent cell get converted into haploid daughter cells. But meiosis 2 is exactly similar to that of mitosis, and it consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. After completion of telophase 2, the 4 daughter cells are formed, which are having haploid, that is single set of chromosomes. We will be discussing all these steps in detail in the next lecture. Have a good day.